All right, everyone, welcome to Chapter 9. This is microbial genetics. I would say this is one of my favorite chapters. I teach genetics on campus. It's one of my favorite subjects, and I did a lot of molecular genetic work when I was doing research. So here's what we're going to do. Introduction to genetics, and I'm going to give you a background of DNA replication. We'll go through transcription, translation, basics of protein synthesis, DNA recombination, and then mutations. All of this, in some respects, should be reviewed from Cell and Molecular Biology, which for the nurses was Bio 1341, I believe. Okay, so let's dive in here. Section 9.1, Introduction to Genetics. So genetics in a really basic form is just the study of inheritance, or you might say heredity, that explores passing your traits from your one generation to the next, right? On another level, we can really look deeper at the expression and the variation of those traits or those genes. We can look at the structure and function of those genes, and then we can look at it how it changes or evolves over time. So there's lots of different types of genetics. You're going to hear the word genome. This is the sum of all the genetic material of an organism. And this is mostly in the form of chromosomes. I will say, though, there is some genetic material that is not chromosomal, and it's not part of what we would call the genome. Okay, so bacteria in particular have a structure called a plasmid. It's extra pieces of DNA. Okay, they're tiny little circles. They don't necessarily contribute. They don't have to be, they're not part of the genomic DNA. And they don't need these to survive. They can copy them. They can share them. Some bacteria have genes for antibiotic resistance, and then they can share it with another bacteria through conjugation. Another source where we see DNA would be in an organelle, such as a mitochondria or a chloroplast, which would be in animal cells and plant cells. These are not genomic either, okay? So these are DNA segments that are used just for those organelles. They make their own proteins. Okay, so that's kind of something interesting. And so genomics is just when you're studying an organism's entire genome. For us, in a human sense, that would be we have 23 pairs of chromosomes, or 46 total in every cell that has a nucleus. And ours are linear. Okay? They look like this. Remember, you have a pair of the first chromosome, one from your mom and one from your dad. It's very similar in bacteria, except for most of the time they have one big circle. This one's kind of wrapped in there, but it would be one big piece of circular DNA. We have learned over time, and in finding new bacterial species, some have more than one circular piece of DNA, so that's cool. And then there's even some bacteria that have linear pieces of chromosomes, which is really neat. Whether you're a bacteria or you're a human, we have double-stranded DNA. And our DNA is wrapped around proteins. That's what a chromosome essentially is. When we take this long strand of our polynucleotides and we wrap it up really tight around proteins, DNA has a negative charge from the phosphate backbone, which I'll show you a picture of, versus the proteins called histones in us and a histone-like protein in bacteria that are positive charge. So they wrap up super tight around that through the charge interaction, and then you can fit a whole bunch of DNA into a very small package. So let's talk about genes. You have this long stretch of DNA. It's just a series of these nucleotides linked together through phosphodiester bonds. So once there's different segments of that DNA that are going to code for a protein. They have a start site and they have a stop site, and we would call that section of that DNA a gene. And when people first started looking into DNA and examining it and experimenting with it, they said, okay, this one stretch of DNA, we're going to call this a gene, a functional unit of heredity. It codes for one protein. And that's what they really thought. Every protein had one gene. And now we know that's not true. The reason is we sequenced the human genome. We're very complex organisms. We would have thought that we would have a lot of genes. So we would have so many genes. But when the sequencing information was coming in, that number was dropping. It wasn't at, 200, 300,000. It was dropping down to 50,000, 40,000, 30,000, until somewhere around 23,000 is what we have. So we couldn't understand how this very complex Homo sapiens doesn't have all these genes. And there are an Easter lily, a lungfish. They have more genes than we do, and they're much less complex. Well, it turns out we have genes that can be read different ways to make different proteins. So that's where the complexity comes in for us. There's another part that changed right here. We now know that not all DNA codes for a protein. Sometimes it can code for an RNA that has a very important function for gene regulation. So that's all new. And when I say new, like 20, 30 years new. Okay, so structural genes, those are the ones that code for a protein. There are genes that code for RNA that's used to help us make proteins. But then there's this class of regulatory genes. They control gene expression, and those are the ones we're really learning about today. A couple of terms you need to know. You've heard of genotype and phenotype, so this should be a reminder. Remember, a genotype is specifically what versions of that gene an individual has. So for humans, for example, we have 2 copies of every gene because you have 2 pairs of every chromosome, one from your mom and one from your dad. So we know that we would call different versions of a gene an allele. For example, if you had a gene for flower color, you might have an allele for a blue flower, or an allele for a pink flower, or purple or orange. You could have multiple alleles. So a genotype would be which specific alleles you have. For a person, let's just say, now this is not how hair color works, okay? Hair color has multiple genes that contribute, but let's just pretend if we said the alleles for hair were brown and blonde, if an individual had 2 alleles and each version of the gene was the blonde allele, which I'm going to use a small lowercase b, right, that would be their genotype. It's 2 blonde alleles. If a person had a brown allele and a blonde allele, this would be their genotype. We're going to say the brown is capital B. So this is their genotype. They have 1 allele for brown and 1 for blonde. The phenotype is the physical expression of that genotype. We often use an example with color or shape because that helps us physically see it, but it's not always that clear cut. In this case, the phenotype would be the person with 2 alleles for blonde hair will have blonde hair. The person that has 2 alleles for blonde and then brown ends up phenotypically looking like brown hair. But that person could pass on the blonde allele and have a baby with blonde hair. So that's the definitions. And this gets back to the size and packaging of genomes. I've already discussed this, but to give you an idea, for E. coli, they have 1 big circular chromosome. It has 4,000 something genes on there. And, if you unwound it, that's a really long piece of DNA. It's, in fact, 1,000 times longer than the actual cell. And I already told you we have about 23,000 genes on 46 chromosomes. So again, how did we do that? I was telling you we package it around those positive proteins called histones. And as a side note down here, you don't have to memorize these numbers, okay? I just put them in there so you understand how it's really packaged in here. This is E. coli. And it's under some experimental conditions where they had the DNA spill out of the cell. This is a great shot really showing you how much DNA is inside of this and how it packages up really tight. So that's cool. All right, switching gears, let's get into the structure of DNA. This is so important, you guys. I just can't stress enough. Structure tells us function. We must understand the structure of our molecules to know how it works. So I really want you to know this and understand this and see how the pieces fit together. If you don't know the structure, you will struggle when we get to replication and transcription and translation. So spend some time really getting comfortable with this. Okay, if we look right here, this is a whole strand of DNA, 1 strand, a single strand, and it's showing you the linkage of nucleotides. So a strand of DNA is called a polynucleotide, but it's composed of monomer units, repeating units called right here nucleotide. So you should absolutely know what are the 3 components that make 1 nucleotide. Okay, this is what you need. This is your phosphate group, and I told you DNA has a negative charge. It's because of these 2 negatives right here on the phosphate group. That phosphate group is hooked to a sugar. Okay, in this case, remember sugars are carbohydrates. So this sugar is ribose, right? And it looks like this. And if you recall, this is a carbon ring. Okay? If they don't show you the letter, that means there's a carbon in there. Otherwise, they're going to show you an O for an oxygen or an N for nitrogen. But otherwise, you always assume like that there's an oxygen or a carbon in there. Okay, and each point is a carbon. Okay, so this is a pentose sugar, meaning there's pent means 5. So there's a carbon here that's number 1, number 2. This is the 3' carbon. 4th one is here, and this is the 5th one sitting up here. And those numbers are going to become important. This section here is a nitrogen base. This is just a cartoon, so they didn't put the nitrogens in there, but honestly, if this were a different book, they would do that. Which means there's actual nitrogen. Sorry, I keep losing my mouse every time on here. In there. Okay, so it's a ring structure composed of nitrogen. Okay, so we need these 3 things. Every single nucleotide, DNA nucleotide and RNA nucleotide, is going to have a phosphate on one of these sugars. Right? If this was RNA, this is ribose. If it's DNA, it's deoxygenated ribose. And that's important. But what makes each nucleotide different is which base they have. Okay, because they all have the phosphate and they all have the sugar. They all have a different base. So if you guys all had looked exactly the same and the only thing different about you was your color shirt, and let's say there's 4 colors of red, yellow, green, and blue, I'm going to classify you according to the color shirt. That's what we do with these bases or these nucleotides. Whenever we're referring to a nucleotide, we just call it by the base. Okay, which is going to be adenine, thymine, guanine, or cytosine, because this part is the same for all of them. That's how we define them, is which base do they have. So let's look at the bases, these nitrogen bases. And here you're seeing, this is more accurately what I'm saying when you don't see the carbons in there, but you see the nitrogens. So we have 4 bases: thymine, adenine, cytosine, and guanine. You absolutely need to know these, okay? The purine structures are adenine and guanine. And I'm going to show you these. They're 2 rings attached to each other. All these lines are showing you covalent bonds. This is a double bond, single bond. So here's adenine. Do you see kind of the 2 ring structures here? And then here's guanine. It also has 2 ring structures here. You do not have to memorize this structure. I'm not going to show you a structure and make you identify it as adenine or guanine. If I had you for biology, I would do that, but I don't get to torture you as much in this class. Okay, so it's 2 rings. That's purines. So that's what defines purines, as having these 2 types of ring structures covalently bonded together. There are other types of purines, okay, but this is in DNA. This is what we have. In the other 2, which are, and we're in DNA, so we're just going to focus on DNA right now, cytosine and thymine. They have just 1 ring. Okay, so this is a thymine here, and this is a cytosine here. And an easy way to remember this, I have to explain that there is another pyrimidine. It's called uracil, and it's found in RNA. Okay, so nucleotides of RNA have uracil instead of thymine. So an easy way to remember, how do I know who's a purine and who's a pyrimidine? Think of eating a pie, like an apple pie, okay? That's how you say this word, so you have to remember how to say pyrimidines. If you're going to have some pie, you have to cut the pie, right? Well, that's how you spell these 3, C-U-T. So that will help you remember which bases are pyrimidines, the ones that are 1 circle like a pie, which you're going to cut to eat. C-U-T, cytosine, thymine, and uracil. That'll help you. Okay, so that's part 1, is just understanding the structure of the bases. They're 1 ring or they're 2. Now the second part of this is our DNA is not 1 single strand. It's 2 strands. Okay, on 1 strand we have all covalent bonds, and on the other strand we have all covalent bonds. But in between the strands, okay, the bases in between have these dashed lines of hydrogen bonds. So I'm going to back up and show you. This is 1 strand of DNA, and we call this our phosphate and our sugar. It's literally called the sugar-phosphate backbone, because this is sort of the side, and the bases stick into the middle. If there was a second strand here, I would have another sugar-phosphate backbone and all the bases sticking into the center. Think of it like a ladder. This is the side of the ladder, this is the side of the ladder, and then these bases coming in the center are the rungs you would climb up. Okay, so the way that these connect together through these hydrogen bonds. We will always end up having a pyrimidine with a purine. Okay, so the number of the rings is 1, 2, 3. 1, 2, 3. You're never going to have adenine base pair to guanine because then you'd have 4 rings. That's too bulky. You're not going to have thymine pair with cytosine. That's too small. That would disrupt the helical structure of our DNA. So you always have to have 3 rings together. So thymine always base pairs, that's what we call it, complementary base pairs, through adenine because hydrogen bonds form here, and it has 2 bonds. That's why I have 2 lines here. I know that kind of looks like the structure for covalent bonds, but I'm just trying to highlight that there's 2 bonds there. And then here's cytosine to guanine, which has 3 bonds. That's important that you understand that and know the difference because it's easier to pull apart adenine and thymine. It doesn't take as much energy to break 2 bonds as it does to break 3. So for example, if you're working in a laboratory and you have to do a PCR reaction, which is polymerase chain reaction where you're replicating and amplifying your DNA, you have to pull the DNA strands apart, okay, and then you're going to replicate them. So you need to know the melting temperature. How much heat do you need to pull the bonds apart? So a lot of times what you need to know is the Cg content. Okay, so, and this is just sort of extra information for you, but you're also going to see this is going to become important when we get to our replication. Okay, so make sure you know how many bonds and who is base pairing to who. That is essential you know that. Okay, so let's look at some DNA structures. We're not going to go over the phosphate because it's just a phosphate group. Just know it's negative. Okay, here's our 2 strands of DNA. So again, here's a phosphate, deoxyribose, and the base. So you're seeing again how these are all the same. What defines that nucleotide is the base, the base, the base. It's different. You can see a purine 2 rings, thymine 1 ring. And this is the second strand. And again, you're seeing sugar phosphate backbone and then the bases. Do you see how it looks like a ladder with the 2 sides and the rungs in the center? And here's 2 bonds, 3 hydrogen bonds, 2 hydrogen bonds. Okay. What else to point out here? 3 rings across. 1, 2, 3, 1, 2, 3, 1, 2, 3. Okay, so always have a purine to a pyrimidine. The other thing to point out, okay, in DNA, we said you are going to have thymine, and it base pairs to adenine. We're going to go back over this when we get to the RNA section, but RNA, adenine would base pair to uracil. And which I don't have featured here, but you'll see uracil. In DNA, this is the ribose sugar. Okay, that's this right here in yellow. It right here on the 2' prime carbon doesn't have an oxygen. Right? This is called a hydroxyl group. It doesn't have a hydroxyl group. It has a hydrogen. So we call this deoxygenated ribose. De means without. So we're saying without oxygen ribose. Ribose in RNA is just regular old ribose, and so you're seeing it has an oxygen here. So there's 2 ways to know if you're looking at DNA or you're looking at RNA. There's actually 3 ways. RNA is single-stranded. That's a big one. It still has a helix. DNA is double-stranded. DNA has thymine. RNA has uracil. DNA has deoxygenated ribose. RNA has ribose. Okay, so this is one way to look at the DNA. This is the helical structure. Here's another cartoony picture. Let me just see here. Let me jump down. Okay, yes, this is what we're getting into next, is this 5 to 3 prime, sorry, there's a truck going by, 5 to 3 prime orientation of DNA. You're going to hear that the 2 strands of DNA run anti-parallel. Okay, anti-parallel. What the heck does that mean? Now you know the word parallel. That means they're running side by side. Okay, so that's that part. The anti means they're running in opposite directions. So here I have a nucleotide. I want to remind you the importance that here's this 5' carbon. Let's start over here. This was number 1, number 2, number 3, number 4, number 5. So we just say 5' and 3'. That's just kind of the terminology. We're concerned about these because this is how our DNA nucleotides are going to link together through these phosphodiester bonds. Okay, so if I was looking at this cartoon, this point right here is the 5' carbon, and this down here is the 3' carbon. So when I'm looking at this one strand of DNA, come on, mouse. Okay, so here's a nucleotide, here's a nucleotide, here's a nucleotide. This right here is the phosphodiester bond. Okay, so the bond is going to form between nucleotide number 2's phosphate to this 3' carbon. So when we look at the orientation of each nucleotide, if we were reading this, we would say we're going in the 5 to 3' direction, because the 5' carbon is sitting up and 3 is below it within each nucleotide. Okay, I'm going to go 5, 3, 5, 3, 5, 3. Okay, now let's look at the other side. It's running upside down. So look at where the oxygen is here. That's your top of your ribose. Do you see how it's pointing down? That's because this strand is running 3 to 5'. Right? So if I start down here, remember here's one nucleotide, a phosphate, deoxyribose, and the base. So this is in one nucleotide, this is the second nucleotide, and this is the third nucleotide. This phosphate belongs to somebody else. Okay? Here's the bond right here. This is our 5' carbon. This would be 4. This is 3. So here's the phosphodiester bond linking this to the next nucleotide. So when I'm looking at the orientation of my nucleotide, 3' is above 5. Okay, so I'm going 3, 5, 3, 5, 3, 5. So this one is running 3 to 5'. So this side of the strand is upside down. Does that make sense, everybody? I know you can't answer me. I hope that made sense, because that gets really important. This picture's trying to show you, but honestly, I don't think they did a great job with the cartoon. I think they should have had the point facing the other way. That's why I put this one in here, even though it's a little blurry. Okay. Here's another picture, and this one's really good because, look, it has the numbers in there on your deoxyribose. So you can really see it a little better. Remember, these are all covalent bonds in this one nucleotide. These are hydrogen bonds in here. They should have done dash lines. This one did dash lines, which is more appropriate. Okay, so here is 5 carbon, 5' to 3. 5, 3, 5, 3. And this one is great. You can really see it upside down. Even the letters are upside down. Okay, so 3, 5. 3 to 5. So again, this is one nucleotide on this side, a phosphate, a ribose, deoxyribose, and a base. Phosphate, sugar, base. Phosphate, sugar, base. This goes to another nucleotide. Why is this important? One, it's structure. Why do we care about structure? If you're yelling function, good. That's right. DNA replication can only move in the 5 to 3' direction. What the heck does that mean? Okay, what that means is there is an enzyme that facilitates making DNA. It's called DNA polymerase. It makes a polymer of DNA. A polymer is just a repeating unit. What's our repeating unit? Nucleotides. Okay, so we're going to take a nucleotide and add it on to a whole strand of nucleotides, making a polynucleotide, which is our DNA strand. DNA polymerase is very specific. It needs to recognize this hydroxyl group coming off of our 3' carbon. So it recognizes this structure, our 3' carbon in this hydroxyl. This is our new nucleotide. It comes in. It has a whole bunch of phosphates. These get cut off. And a bond forms right here between this oxygen and this phosphate. Okay, and that's how we're going to put this new strand on. So if you are this enzyme, DNA polymerase, the only way you can build is if you attach, come on mouse, right here. That's why we're going to say it's in the 5 to 3' direction, because when the new nucleotide right down here, if I could ever work this mouse, is going to be, you can't really see the 5', but the 5' would be here, and the 3' would be here. Okay, the orientation that it gets put in, the new nucleotide comes in the direction of 5', 3, 5', 3, 5', 3. Okay, so again, DNA polymerase has to attach right here to put in a new nucleotide, and when it orients that new nucleotide goes 5', 3. So this whole strand is going to be in the direction of 5', 3, 5', 3, 5', 3, all the way down. So we say DNA replication goes in the 5' to 3' direction. Okay, now you still have to remember, this is what DNA polymerase is recognizing. Okay, it cannot put, I know they put the 5' here, but the 5' is actually underneath this. Okay, if you remember, it's down here. It can't attach over here to a phosphate group. Okay, DNA polymerase can't attach to phosphate. It can't do that. If I come back here, that would be attaching up here somewhere. It can't do that. It needs this structure right here to attach to. Okay, I hope that all made sense. Okay, here's some DNA to RNA, and we're going to revisit that, okay? Here's our bases again. Cytosine and thymine, remember, one ring, because like a pie. And then here's our purine and our adenine, which are, these are our pyrimidines, these are our purines. And you can see the color bases all matching up. And this is RNA, one strand, and now we have uracil instead of thymine. Are you ready for some DNA replication? It's the type of replication, we call this process semi-conservative. And this is true for all organisms that have DNA that replicates. Okay, it's semi-conservative because we're going to conserve half of the original parent strand. Okay, so semi meaning half. Our parent strand is the original DNA molecule that we're going to copy. So we call it the parent strand, and we call the new strands that we make daughter strands. That's just the lingo. I don't know. Scientists. Okay, so this is our dark blue parent strand. You can see the pairing between the bases. Remember, these are hydrogen bonds. So we're going to pull this apart. Okay, we're going to use enzymes to pull this apart. And that's the separation of the strands. So there's our parent strands. This is why Mother Nature is so cool. There's many reasons. This is one. DNA replication is so simple. Okay, it's so simple. You have 4 bases. You can't get more simple than that. But the complexity is in the sequence. You can change those into any sequence difference you want and get all different kinds of proteins. Okay, which are DNA is the code for proteins. We'll get there, but if I have just one strand of DNA, I automatically know the second strand because of complementary base pairing. I know that if there's an adenine, I put in a thymine. Cytosine gets a guanine. Thymine gets an adenine all the way down. So if you have one side of the DNA, you automatically know the second side. So these are all new nucleotides being attached in. These are going to have covalent bonds here, here, here. The nucleotides, remember, getting covalent bonds. The hydrogen bonding is what does the helix. Okay, so that's going to happen on both sides, and you're going to end up with 2 brand new, this is a double helix, and that's a double helix. It's semi-conserved because you still have half of the parent strand and you have half new nucleotides. Okay, and here's that word, phosphodiester bonds, if you don't remember that from biology, but that's between the nucleotides in one strand. Okay, it's between the sugar phosphate backbones. Remember, in between the strands is hydrogen bonds. So this is what DNA replication looks like this could be a linear piece of DNA, but it also looks like this in the circular bacterial DNA. You're going to pull apart this DNA. Here's your DNA. Here's the 2 strands. They're purple or blue and red here, but here the parent is blue and blue. Okay, so you can see it right here. So it's like you just took this and broke these bonds and pulled it apart and made a bubble. Do you see that? Like if this part wasn't in there, you would just have straight blue tracks, right? But we pulled these 2 parent strands apart and we created what's called the replication bubble. We do this at the point of origin. And this is a section where there's mostly in bacteria, they have this actual specific spot in the chromosome, the circular piece of chromosome that is AT rich. What does that mean? That means that a lot of these base pairs are adenine, thymine. Because remember, how many hydrogen bonds did they have? 2. Okay, so it's not very hard to pull those apart. You don't need a lot of energy. So this part can pull apart really easily. That's going to be our origin and form this bubble. When we go to study this, we're going to just ignore half of the bubble. We're going to ignore one half and just look at one replication fork. Okay, so this kind of looks like a fork in the road. Okay, and DNA replication is moving down this way. So one strand, we're going to call the leading strand, is going to keep going, copying this parent. The other half of the fork is called the lagging strand, and it actually goes the opposite direction. So it has to lag behind. The DNA is directional. It only goes 5 to 3 prime. This one can just go right through 5 to 3 all the way down. But this side will be running opposite. I'm going to show you examples with all the numbers in there. So it has to go this section first, and then this has to pull apart. This one would go straight up, but then you would have to jump up here and do this little section. Okay, so I'm going to show you that when we get through. Let's stop. Let me see where we're at. Where's my Kaltura? Okay, I'm going to pause this, and we're going to go to the next video, okay? Or I'm going to finish this one and start. DNA replication will be its own video.